I have been involved with, and it's great so early in the morning to be so enthusiastic about this topic. There's been a lot of comments that people have said, yes, I will address that. I hope I can meet all those expectations. And I think most of that will come in the discussion period. In this presentation, what I was asked to do is talk about how is the safety assessments of low-calorie sweeteners conducted, and what is the overall global approval process. So I won't specifically be talking about the individual sweeteners, but I'm very happy to elaborate on that if we have time as we go through. I think we should skip lunch, actually, and just keep going. Okay, so as we've heard already, the use of these has traditionally been to reduce the caloric content of foods and to reduce the glucose content so that they are suitable for diabetics and blood glucose control. And we'll talk about the controversies about that, but that is traditionally their use. As such, they are defined as a food additive. That automatically means that they go through the traditional food additive risk assessment. That is the process that I'm going to describe for you today. And I do hope that by the end of it, you will be convinced that it is an extremely thorough process. So, as was already mentioned, the first step is that the safety is assessed by an independent scientific body. This assessment must be undertaken and approved before the products can be used in the marketplace. On a global or international basis, that is done by, as was mentioned, JECFA. They are the ones responsible for the safety assessment, and that is the Joint Food Additives Organization Expert Committee from WHO. Then, in addition, there are regional also assessors. In some countries, they simply accept the JECFA and Codex regulations, accept the JECFA conclusions, but in many cases, there is an additional step undertaken in this specific region. So, of course, in Europe, that will be EFSA. In the United States, it's FDA. India, we've already talked about the FDA. Of course, because I'm from Canada, I have to mention Canada. That is done by Health Canada. And in each situation, that is basically the same process that is undertaken, but depending on the different specifics for that country, they may result in a slightly different acceptable daily intake, and I will highlight that in a couple of places where that has occurred. So the role then of the risk assessors is to first and foremost establish the safety of that food additive, and in this case we're talking about the low-calorie sweeteners, then to establish what are the acceptable levels of intake and the acceptable use levels for those food additives. I don't think I'll go through this. It will be, I believe these presentations are going to be made available so you could review them. In terms of JECFA was established in the 1950s and set out the basic principles for evaluation. This is constantly also under review, but the basic approach to how we evaluate the safety of food additives and low-calorie sweeteners was established during that time. And then this again was reviewed to ensure that with change in science, advances in methodology and so on, that it was still a valid approach. It was reviewed, and again, it was just recently updated in 2009. And it does include, this is a very comprehensive review that is publicly available and talks about the very key issues, some of which we have come up with today. What about safety in children? What about pregnant women? What about specific different types of countries and so on? These are addressed in this extensive review to ensure the process does properly consider those variables. So I think I want to first of all just kind of talk about in a general way, what do we talk about when we're talking about safety and risk? Because the traditional scientific method is saying we're doing a risk assessment. And that basically is to show that something is safe. If it's safe, it seems that is the absence of risk. And we are talking about it today in the context of food additives, but we actually do this with many different aspects of our lives. We talked about car accidents, and so, you know, what is the safety of driving in India? I'd say that may be slightly different than the safety of driving in northern Canada, where there are very few cars. So, you know, and there is also this whole question then, we apply to it not only in food, we apply to it to many different factors, medications, drugs, and different personal choices. And in all cases, the process is the same in that risk is defined by two components. And I think often we forget about both, that there are actually two components. One is the question of hazard. What is the potential of that activity, that compound to actually cause harm is one, but the other is the exposure. So for example, I just gave you the example of driving. I mean, the difference in the risk of a car accident in India versus Canada is the exposure to other cars. In the situation of food additives, it is the exposure of how much are we actually going to consume. So whenever we talk about something is safe, you cannot just say it is safe no matter what. It is always, is it safe under the intended use, under the likely exposures, and that must be considered. So let's talk a little bit about, you know, hazard. So what is the potential to cause harm? I just picked up two compounds here that I think most people are familiar with. So if I said, which is more hazardous, more likely to cause harm? How many would choose the cyanide? Okay, how many would choose the water? Okay, most of you didn't vote, right? So that's the problem. Obviously, most people would say, you know, just from history of use and personal use, most of you every day would drink water and have no worries about it. If I handed you a bottle of cyanide, may have a little hesitancy before you chug that down. So there is an inherent difference just in terms of the potential to cause harm. And that would be what we would consider. How would we define? How much does it take to cause harm? Is an identification of the hazard. The thing is, we can accept and have an acceptable level of risk, one that does not permanently cause harm or have a detectable level of harm if we have a low enough exposure. So here again, let's talk about our cyanide. We all know about that we can have cyanide poisoning. Did you know that I would say most of you, even today on the breakfast table, there was a source of cyanide? And I think many of you have eaten cyanide. That source is almonds. Cyanide is actually present in many different foods. You can eat it every day, a small amount. Your body very rapidly metabolizes it, and you have no problem with it. It's a matter of dose. I wouldn't recommend popping open the bottle and chugging it down, but definitely low levels, intermittently, causes you no harm, and you all have long history of use of cyanide, actually, whenever you eat almonds. On the other situation, if you have something that traditionally has a very low level of hazard, like our water bottle, you can tolerate a much higher level of exposure. So let's just kind of do, again, a little question here. This is a 500 ml bottle of water. Everybody has one on their table, probably would have no concerns drinking this entire bottle within an hour. How many bottles of water can you safely drink in 1 hour? Any guesses? Well, I'll let you ponder that, and I want you to think about it because it's going to come into this question of what is the amount, what is the safety factors? How high should we be willing to accept? Is it okay? Is this still considered safe if 5 bottles of water in an hour is okay? Is 10 bottles in an hour okay? What is the level of safety that we are comfortable with? This was mentioned before, but it's very important, and especially as a toxicologist, I am so aware of this, and I think it is one of the biggest misconceptions that there is some difference between whether something is natural and man-made as far as whether that defines its hazard. It absolutely does not. For every chemical, whether it's a chemical in food that's naturally occurring or whether it's a man-made chemical, there's always a dose that is going to cause toxicity. In fact, we're going to later this afternoon have a discussion about nutrients. I would say that there is, for many of the nutrients, there is a level of toxicity, and it's often not that much higher than the level that is required for health. And for pretty much everything, there is a level that is safe, that will not cause an adverse effect. We can debate that. There are some things in terms of potent carcinogens and so on, which we accept. Pretty much we would not say there is a safe level. But for mostly across the board, we can define a level that is safe. It may be very, very small, but there will be some level. And this is the kind of graph that we need to understand. This is a dose response. So with increasing dose, there is going to be a region of no effect. For some compounds like vitamins and minerals, going to get a region of toxicity. So that is what I mean. You do not say that blankly something is safe or not. It is safe within the region of no effect. It is not safe within the region of toxicity. At some point, you will get there. The rate of consumption is also important. So I just said, in terms of drinking the water, we all would accept you could drink, a certain amount, that 10 bottles of water easily over the course of 2 or 3 days. If we start talking about lifetime exposures, then we're talking about, again, a different amount. That is a very different exposure to trying to consume something all at once. And that, when we're talking about food additives, that is an important distinction. We are expecting that you're going to be exposed to them a small amount, but potentially a small amount every day for the rest of your life, the type of safety we are looking at. And what you will see is that, this results in a very different level that is actually going to be in the tissues. So if you take something, whether we're talking about a food additive, whether we're talking about calcium or iron, taking a very large amount, you're going to have a rapid level of absorption, reach high levels of tissue concentration, and then slowly metabolize and excrete it. This is very different from taking that same amount, just a small amount every day, where between each dose, you have the opportunity to metabolize it and excrete it. That is why we can eat almonds every day, because you're going to quickly metabolize and excrete it, and next day you're fine to go again. So again, you have to put that in consideration. And experiments on food additives, which take the first approach and look at a very large amount over a very short period of time, do not represent what is going to happen in the second scenario, which is typical for human consumption. So we must take that into consideration as well when you're looking at some of the studies. And the gut microbiome is going to be one where I'm going to talk about that. Because of this very, trend that we need to drink more water, and water is good for you, and rehydration is critical, people can and do drink themselves, sadly, to death. And athletes, there was a woman very recently that in some contest, to see how much water she could drink. So that is the amount, 6 liters for a typical person. I think that's actually, so we'll say that's maybe 10 of these bottles, right? So again, I just kind of want you to put that into your mind. So let's now talk more specifically about the hazard evaluation of food additives. An extensive number of tests is required. Sorry, I'm getting worked up here, getting a little warm. It actually takes years to conduct. The simple, in sort of the straightforward way, the objective of these tests are to answer the following questions. What happens to that compound when we consume it? Do we absorb it? Does it simply go right through us and be excreted in the feces? What is the toxicity following long-term consumption? We are very much moving away from any short-term, high-level acute tests for food additives because those results are not even meaningful. What is meaningful are long-term, just as the question is, what about consuming it for life? What are the effects there? Of course, any effect on mutations or cancer development, and I'll say that that is a critical question, that there be no impact on the DNA, no evidence of cancer development in order for the food additive to be approved. The other question is potential impact on reproductive toxicity. That means, does it impair the ability to get pregnant, both male and female? Does it impair the success of the pregnancy coming to term? Does it impair any type of outcome of the pregnancy or survival? And all tests to confirm that there is no effect on reproduction or development of offspring. Because the sweeteners are specifically targeted for use by diabetics in order to help them control blood glucose, human clinical studies have been done on every one of the approved sweeteners in diabetic individuals to confirm they do not impact blood sugar and they do not impact insulin release. So those clinical studies have been done for all of the approved sweeteners as well. So in terms of then the overall risk as the process, just to lay this out, the first one is, as I just said, using the toxicology studies, identify what is the hazards, what are the dose response curves, and from there we will, the regulatory agencies will establish the acceptable daily intake. This has been mentioned before and it's very well described in the handout that you received, but I am going to go through it because I think it is critically important that you understand it, and I'm actually going to disagree slightly with the previous speaker in terms of how it is calculated. The other is then, once you have determined what is the amount you can consume every day, the other part is to then assess what is going to be the likely exposure to individuals depending on how much are added to the different food categories as were described. There's so much allowed in different food categories. What is going to be the ultimate expected intake? And the final aspect then is sort of characterizing the risk. We need to determine, do any of the proposed uses of these ingredients in different food categories, in the dairy products, in the sweets, in the beverages, result in intakes that exceed the acceptable daily intake for anybody, any sex, any age in the population? And the answer must be no. It must be no, or else the level that is approved or can be allowed in those different food categories must be reduced. So this is actually, in my experience, most often where you have somebody exceeding the acceptable daily use is in children. If you are trying to put a food additive into a food product that is consumed by children, such as a fruit juice where they tend to drink much more per body weight, and often children kind of get into these habits where they want that same juice every day for the 2 months, and you can't get them to switch. I have a 2-year-old like that for every day wanted the same food, and you're like, how can you eat that? But those are the types of behaviors that mean that children are often most susceptible. So what will happen then is the amount that is allowed in that food product must be reduced in order to ensure that even for children, the acceptable daily intake will not be exceeded. So we talked about then establishing this dose response. So there are very well-established international protocols and requirements for how these tests are done. They outline specifically the number of animals needed for adequate statistical power for the studies, the length of the experiment, the specific experimental conditions, the type of diet, the purity of the compound, and so on. And the list of parameters that must be monitored for these regulatory submissions is extremely extensive. Every animal must be monitored for how much food and water do they eat. What is their body weight changes over time? Experiments such as the reproductive toxicology tests, there may be additional specific experiments or parameters that are evaluated. Now, this is really important because it is quite different from often what we read in the published literature. I have evaluated and have reviewed hundreds of studies on low-calorie sweeteners. 2007, I published a review on aspartame. There were over 700 papers at that time. It's a lot of work, and it took me a long time. The point is that in many of those studies that are actually published in peer-reviewed journals, most of this, or a number of these parameters are not reported. And this is true, I'm going to show you later, for the gut microflora study, where a lot of this information is not there and makes it very difficult to actually determine whether or not it is actually truly the compound that is causing the effect, or is there some other parameter, such as change in food intake, that is having the effect that you're seeing. The whole objective of all of these studies is to establish the no observed adverse effect level. This is the level that can be consumed every day with no adverse effect. So this is a little different from what was said before, but the acceptable daily intake is not based on the level that shows an effect. The toxicity level is up here somewhere. The acceptable daily intake is based on establishing the level that caused no effect for consuming every day for the majority of the lifetime. That is the no observed adverse effect level. So the acceptable daily intake then starts with that. It doesn't start with the toxic level. You must establish a level which, fed for the lifetime, causes no effect. And that is, again, done in multiple species, and you use the most sensitive species. Then you divide that by a factor of 100. That is called the safety factor. That is to account for potential differences from young to old, between individuals, and any potential differences between humans and the animal species. I must tell you, though, that there is also specific studies done to ensure that the animal model does adequately represent the human model. So for all of these sweeteners, the metabolism, the rate of absorption, the rate of metabolism and excretion has been evaluated in animals and in humans. Intake does build that in for additional conservatism. So what that means then is if you have a level, and this example is from aspartame, the no observed adverse effect level is 4,000 milligrams per kilogram body weight per day, or 4 grams for every kilogram per body weight per day. The adverse effect level, where there actually causes some toxicity, is upwards of around 10 to 12. And the adverse effect there that is seen is growth inhibition, because at that point you have an imbalance in nutrition. Once you start getting greater than 5 grams per kilogram body weight, you start to have an imbalance in nutrition just due to how much of an additive. The no effect level, 4 grams, you divide that by 100, you're down to 40 milligrams per kilogram body weight per day. So it is very conservative. It's not starting with a level that causes an effect. It's starting with a level that causes no effect, and then it is divided by 100. So just think about that before, too, when we were talking about if we needed a 100 safety fold factor for the water. you would not be able to drink very much water every day if you actually applied that 100 safety folder. In fact, you would not be able to eat very much of anything if we started to apply a 100-fold safety factor to everything in food. Again, just to put that into a graphic illustration, the level with no effect in a lifetime divided by 100-fold gives you the acceptable daily intake, safe to consume every day for life. Okay, so what about the children? So there's a difference between infants and children. Infants, from a regulatory standpoint, from JECFA, is defined as birth to 12 months of age. Children are 1 to 12 years of age. Are these adequately protected by the acceptable daily intake? It's a very valid question because, as I said, infants and children tend to be more sensitive. They tend to eat more per body weight. There is the potential that they differ in their capacity to detoxify and eliminate compounds. They are still under development. And because they are still in development, they are potentially more susceptible to toxicity. So that is considered. It is considered from the point of view both within the safety factors and also from the point of view that any effect is during growth and development is taken into, is evaluated during the toxicity testing because the animals are given the food additive from weaning. And in the reproductive studies, they're given them from in utero through 3 generations. So if that compound actually affected any growth or development, that would be shown, that would be considered in the no observed effect level. If there was an observed effect level in that experiment, you'd have to come down to a level in that experiment that had no effect on growth and development. So So the variation then is addressed both within the 100-fold safety factors and with the studies that look at any impact during growth and development. And as I've said, there's also been many studies actually done in human children as well. So generally, the acceptable daily intake is considered to not apply to infants 12 months and younger, and those require specific different evaluations for products that are going to go into infant formulas. But it does apply to children. So I strongly agree that at this point there really is, from an international perspective, an understanding of the acceptable daily intake that it is to be inclusive of children. Pregnant women, same in terms of reproductive studies to cover that question. And if there is any effects, it will be established and reflected in the no observed effect level. So the acceptable daily intake does also protect pregnant women and is applicable also for pregnant women. So again, in terms of the safety of food, these low-calorie ingredients, they are safe to consume by pregnant women as long as it is below the acceptable daily intake. So just briefly, the whole question of how do you assess exposure is an entire different topic, but basically it is the maximum permissible use levels that were discussed earlier are set to ensure that the exposures, the intakes by the population will not exceed the acceptable daily intake. And again, there's a number of different sources of data that can be used, disappearance data, how much food is actually entering the food chain, the categories such as by codex that are established, different surveys, and then of course there is also can be dietary studies that are done as well. So again, just highlighting then the whole objective is to ensure that the exposure is not greater than the acceptable daily intake. So let's now move a little bit more specifically to the low-calorie sweeteners, that all of the sweeteners currently approved have been thoroughly tested and acceptable daily intakes established. Just a note again in terms of the sweeteners and the use of mixtures. That is actually, from a toxicology standpoint, a very good thing because the greater variety of different sweeteners that are in the marketplace lowers the likelihood that you are going to overconsume any one in particular. So for example, if you have your aspartame and ACE-K in your beverages, you use your sucralose in your high processed, high heat processed foods, and you put it also in your tea, you're basically going to be reducing your exposure to each individual one. And so it lowers your risk of exceeding the acceptable daily intake. Just to point out how much actually, we consume. Just to replace, I think this is interesting in terms of putting into context, to replace the 25 grams of sugar. We are talking here in terms of, between 40. Sucralose you need the least because it has 60 times the sweetening potency of sugar versus the other ones, which are around 200. I think I want to mention that it is inaccurate, actually, to say that these are sweeter than sugar. They are on a gram per gram basis. They have a greater sweetening potency, which means you can use a very small amount to replace sugar. But you actually cannot achieve the same level of sweetness that you can with sugar. So if you want something really, really sweet, sugar, the higher the concentration, you can reach a higher level of sweetness than you can with the low-calorie sweeteners. So they're fine for replacing in certain products, but I think some products that are incredibly sweet are very difficult to actually use a sugar replacement for that because you can't get that intense sweetener. And that also comes to the question of impact on the brain. So even though you say they are 200 times the sweetening intensity, you do not take the same amount. You're taking 1/200th. So to say, well, because there's so much sweetener, you get 200 times the impact on the brain, that's forgetting that you're taking 1/200th of it to get the same sweetening level. So it's a nonsensical sort of, people forget, it has a high sweetening intensity, but you don't take very much. So let's put that now the next step. Well, how much do actually people eat? And there are many surveys that have been done on this around the world. And I don't believe, I've only seen one actually for India, but most of them have been done in Europe. This is specifically for aspartame, one of the more commonly used ones in North America. There you have the no observed effect level at 4,000 milligrams per kilogram per day. Acceptable daily intake is 40 by JECFA. In the US, it's actually 50. They have set theirs at 50. The highest users, those that consume many different diet products, tend to come in on average around 15 milligrams per kilogram body weight. The average user, not a high user, is around 2 to 5 milligrams per kilogram. And this is just people that use them. This is not average for the population. This is just average user. So you can see it's actually even much, much lower than the acceptable daily intake, which is much, much lower than the amount that causes no harm in animals, which is much, much lower than the amount that actually causes an adverse effect. So the likelihood of getting an adverse effect at that level is extremely low. Oh, very low is what I said before. Very low level of risk, very high level of safety because of the extremely low levels of use because of the intense sweetening properties. And again, you can look at this for all of them. This is for aspartame, the acceptable daily, the estimated daily intake, around 6% of the ADI. Saccharin is a bit higher. It has a lower acceptable daily intake. Sucralose, again, around 32%. This is actually from the FDA, and I just wanted to highlight, this is a similar table, but the numbers are actually going to be different from what we saw in the previous presentation. Actually, the previous FDA numbers were incorrect in terms of number of servings, of number of packets per day. If you look across the board there, Again, for aspartame, because of the high acceptable daily intake, up to 75 packets of sweetener per day before you reach the acceptable daily intake, up to 18 to 20 sodas, depending on how much is being added to them. So across the board there, you can see it's a pretty high level of intake, especially if you start mixing these up in terms of taking one type of sweetener in one product, and that is very typical because, again, the optimal use of these sweeteners is different in different food products. Aspartame, as we said, does not work well in high heat temperature. It's actually a dipeptide, like a protein, and it breaks down. You all know you cook proteins, they change, and it loses its sweetness. Sucralose is the most heat-stable one and is the one found in most high-temperature applications and in all baked products. So, to conclude then, the safety of the low-calorie sweeteners has been extensively evaluated worldwide. It has been evaluated by JECFA and then again by numerous different agencies subsequent to JECFA evaluation. And of course, by a number of other health professional organizations, as was mentioned in the last presentation. I hope I've convinced you that acceptable daily intake is extremely conservative number. It is based on a level that causes no effect in lifetime and then further reduced by those safety factors. So it does apply to all consumers, excluding infants up to 12 months. And again, over and over again, with every survey that has been done around the world, we are confirming that even the highest users, and the highest users are often diabetics, and sometimes teenagers, especially teenage girls who are really concerned about their weight, will be consuming a lot of diet products. Even those high intense users are highly unlikely to exceed the acceptable daily intake. And also, this is not an unending story. There's not simply an approval and then the regulatory agencies walk away. There is constant re-evaluation. EFSA just did a very thorough re-evaluation of aspartame that was published December 2013. Health Canada just did a very critical re-evaluation of saccharin. And of course, the stevioglycosides continue to be a topic for many regulatory agencies, and because their use is increasing, they are continually being monitored. So it continues to be reviewed, and over again, they confirm the safety of use. And I think I'll stop there. I'll be happy to talk more about the specific sweeteners if we have time. But for now, I think my time's up. Thank you.
Thank you. Thank you very much. After the detailed
You're not on.
And risk assessment. Now it's open for questions. Quickly, please. Yeah.
Lactation, are these sweeteners secreted in breast milk, and since the child is only on breast milk, have the safety issues been assessed over there also?
Perhaps maybe the best way for me to answer that is to just talk very briefly about what happens to each of those sweeteners when we consume them. So may I show some more slides?
Okay.
I have them right here. I'm okay. I have them right here. So, aspartame. Aspartame is actually, as I said, 2 amino acids that it contains a methyl group. So aspartame actually, when it enters the digestive tract, is completely digested down to these 2 amino acids and the methyl group. These are commonly found in foods. So here you can actually see how much phenylalanine and aspartame or aspartic acid would be in a soft drink compared to the amount that is in a glass of milk. So similar also to the methanol, much lower levels than you find in foods. So there's no more likelihood that you're going to change the composition of breast milk from consuming aspartame than from having your glass of orange juice and yogurt in the morning. It is completely digested. It never enters the body. Sucralose is not digested. It is not absorbed. It simply completely goes through the gastrointestinal tract and is excreted in the feces. Because it is not absorbed, it absolutely cannot get into the breast milk. Same with the other ones. Actually, some of them are absorbed. Saccharin is absorbed, and so is ACE-K. But again, the levels are so small, and they are very rapidly removed. So, second, to talk about stevioglycosides.